Let's make it start. A very warm welcome to the third of our ongoing series of Advanced Therapeutic Webinars, with today's webinar focusing on ocular gene and cell therapies, the new frontier of vision, research and treatment. My name's Ian Alexander and I've got the pleasure of chairing the webinar today. For context, I head the Gene Therapy Research Unit at the Children's Medical Research Institute and Children's Hospital at Westmead, and am Director of Laboratory Research at Kids Research, Sydney Children's Hospital Network. I'd like to begin with an acknowledgement of country. So this webinar is being hosted from the traditional lands of the Gadigal and Bidjigal people of the Eora Nation in the east and the Burramadigal people of the Darug Nation in the west of Sydney. We acknowledge the traditional custodians of the various lands on which we are meeting today and the Aboriginal and Torres Strait Islander people participating. We pay our respects to elders past, present and emerging, and celebrate the diversity of Aboriginal peoples and their ongoing cultures and connections to the lands and waters of Australia. So before introducing our speakers, just a few quick housekeeping and structural issues. We have two presentations from excellent speakers today. Each of those will be about 12 to 15 minutes and will run sequentially. And then the remainder of the hour will be question and answers. So we have the video of the webinar will be emailed out to all attendees and available via our website in the coming days. Our Q&A panel will be answering questions, and we encourage people to submit questions throughout the webinar via the Q&A tab in the Zoom controls. And if you see a question that's already been asked that you would like to ask, see asked, then upvote it by the question by clicking on the thumbs up beside it. So we'll also be providing any answers to unanswered questions via our website and emails in the coming days following the webinar. So without further ado, let me introduce our two excellent speakers, Professor Robyn Jamieson and Dr. Anai Gonzalez Cordero, who are leaders in the fields of genetic retinal disease and stem cell biology, respectively. So Robyn, who will be our first speaker, is a physician scientist and heads up the Eye Genetics Research Unit, which is a combined initiative of the Children's Medical Research Institute, Sydney Children's Hospital Network, and the Safe Sight Institute. And she's also led the clinical implementation of the Ocular Gene and Cell Therapy program through Sydney Children's Hospital and New South Wales Health. And Robyn's work has contributed to the recent Therapeutic Goods Administration approval of the first clinical ocular gene therapy in Australia. And I'm sure you'll learn more about that during Robyn's talk. Dr. Cordero is the head of the Stem Cell Medicine Group and Stem Cell and Organoid Facility at Children's Medical Research Institute and the University of Sydney. And Dr. Cordero is a world authority on ocular organoids with past work including the establishment of proof of concept for stem cell therapy by transplantation of photoreceptors as treatment for blindness. Currently at the CMRI, Dr. Cordero's group develops novel technologies to test retinal cell and gene therapies and the modelling of retinal, auditory and brain diseasing using stem cells and organoids. So without further ado, I invite Robyn to speak to us.
Hi, Ian. Thanks very much for that. Thanks very much, Ian. And it's a pleasure to be here. I hope everyone can see that. We're now talking about advanced therapeutics and ocular gene and cell therapies. So to give you an overview, so we're mainly talking here about inherited retinal dystrophies and the new therapies that are becoming available in that field. And the reason that we're excited about this is that for many years, people with these conditions have been going blind and just having progressive vision loss. And there's really been no therapeutic possibilities available. And just a quick introduction to the particular conditions. They often affect the rod photoreceptors and cause a condition called retinitis pigmentosa, or they can affect the cone photoreceptors and cause a condition called cone rod dystrophy. And in the retinitis pigmentosa, you often lose peripheral and night vision first, whereas in the cone dystrophies, you often lose central vision first. But often, and again, depending on the disease process, this can lead to degeneration and then complete blindness. And one of the reasons that this has just been so difficult to tackle, both from a genomic diagnostic point of view and then also from the point of view of potentially thinking about therapies, is that there's a vast amount of genetic heterogeneity. And what that means is that there's over 259 disease genes in these groups of conditions. And as indicated in the diagram there, there's lots of overlap. And so it's hard for an ophthalmologist just to look at the back of someone's retina, back of their eye, and say, oh, this must be due to a mutation in a particular gene. And so it's been very hard before we had genomic approaches to sort of pick the one particular gene that we should look at. And these conditions are relatively common, in quotation marks, for rare diseases. They affect, there's recent genomic data that it's indicating that they affect around about 1 in 1,000 people worldwide. So this is not an insignificant clinical problem. And then the other element about the eye, which makes it suitable for advanced therapeutic approaches, is that it is accessible to therapy. So it's able to be accessed through the front part of the eye, and then genetic therapies can be introduced via a subretinal injection at the back of the eye. So these don't have to be infused through into the blood and possibly have those sorts of systemic side effects. So this is also an advantage in terms of eye genetic therapies. And the other element that's an advantage is that the eye is small. So there's a relatively small amount of the vectors and so on that need to be developed. So that is also an advantage for ocular gene therapy. So this is something that we put together in terms of how we're going to deal with this and what we need, the things that we need to be doing to help our patients who've got their impaired vision from these types of conditions, to get them to a situation where they may be able to be in a vision rescue situation. So there are many elements required along this pathway, and of course in this time I'm not going to speak about exactly every one of them, but I just want to give you an overview and a kind of an outline of where we're heading with this. So for diagnosis and staging, a multidisciplinary team approach is really, really valuable. And I'll talk a little bit more about that because for the full care of the patient and for the full way moving forward towards a therapy, you do need input from the ophthalmology team, from the clinical genetics team, and from the laboratory genomics team. And then, of course, other teams as well as you get more along the therapy pathway. And from this initial assessment in the diagnosis and staging, you're thinking about, well, what is the genetic diagnosis? You're thinking about therapy suitability. And then, as I'll show you, you also need to think about functional genomics, because sometimes these genetic variants are not clear cut. And then the pathways that you're thinking about for the patients, could there potentially be an internationally approved gene therapy? And we've had one of those now approved in Australia for therapy for RPE65 based retinal dystrophy. And then there are others that I'm sure will gradually come online. But you know, that's one pathway that you might want to pursue, if at all possible, a clinically available therapy. Then there are other pathways like clinical trials, which can be genetic therapies or also pharmacological therapies. And there are quite a number of clinical trials available internationally that are happening at various stages of reporting for various types of retinal dystrophies, including those due to mutations in the choroideremia gene, RPGR gene, achromatopsia due to mutations in the CNGB3 gene, and so on. And then there are oral therapies that are undergoing clinical trial as well for conditions like Stargardt and Usher syndrome. And then the other approach that we are working towards taking is investigator-led clinical trials. So novel genetic therapies that we and others may be developing as well. So we're all aiming with these approaches to get to the clinical therapies and the clinical trials for these patients. So then in terms of the diagnosis and staging, so what are the requirements for genetic therapies? And I'll run through this, particularly in relation to the RPE65 gene therapy. So you need the clinical diagnosis, the genetic diagnosis, and the disease staging. Because for a gene therapy, you, generally speaking, need to have viable retinal cells. Because you can see from this diagram down below here that the retina is there's lots of cells in there. And what a gene therapy does is it aims to put in a replacement gene to help then pump out the protein to help those cells function better. But if you don't have any cells, then there's not much, the gene therapy can't really help, generally speaking. And so that's why Anai will be talking subsequently, because that's a new and exciting space in terms of potential cell therapies. So then in terms of talking about where the multidisciplinary team comes into that, so in terms of RPE65 related retinal dystrophy, so the first thing is a clinical diagnosis, a clinical ophthalmic diagnosis. And this might be something like what's called Leber congenital amaurosis, which is a severe retinal dystrophy which presents in the first year of life. Or it might be what's also called an early onset retinal dystrophy, which is in the first 5 years of life. And the patients can have sort of a flecked appearance to their retina, and then they have the layers, you can see these layers in the retina on OCT. I'm not sure if I can get this marker to work, but anyway, layers in the retina on the OCT, and they need to have cells seen here on their retinal OCT to be eligible for therapy. They also need to have a genetic diagnosis, and this is on the basis of the TGA requirements, and I'll put the website address there for the TGA, which requires pathological biallelic RPE65 mutation. So both parents must be shown to be carriers, and so there's a variant from each of the parents. And then in terms of the staging, they need to have sufficient viable retinal cells, and that's on the basis of a clinical assessment, not any one particular test, but a clinical assessment which may include things like the visual acuity, visual fields, the OCT, the ocular coherence tomography, mobility tests, and FST test, again, looking at retinal sensitivity. So those are requirements for that RPE65 gene therapy, and as more of these come online, similar sorts of things are going to be required: the clinical diagnosis, the genetic diagnosis, and then the appropriate clinical staging. So one of the reasons the multidisciplinary team meetings are so valuable in terms of this combination of expertise is that you often need this to be able to come to a clear-cut genetic diagnosis. And so it's very important that ophthalmic phenotyping is done in the patient and often family members for the appropriate test ordering and interpretation. And there's a number of ways that these genomic investigations are done: panels, exomes, genomes. There's a number of approaches. And what often happens is that a particular set of genes is examined. You might have the whole genome there, but you would usually start off looking at a group of genes, like 50 or some amount of genes like that, depending on the particular condition, depending on what the ophthalmic phenotyping is telling you. And that just helps cut down the analysis time for the laboratory and not asking them to be looking at a whole load of irrelevant genes. Of course, you can take this approach, and then if you don't find anything in the first place, well, then you can expand out to other genes and other groups of genes if you will think that there might be a suspicion that it actually could be in another group of potential genes. And then once the result becomes available, it's very important to be sure that the genotype actually fits the phenotype. For instance, we've had cases where there can be a referral and the patient's referred as having retinitis pigmentosa, but actually they're found to have congenital stationary night blindness according to the genetic variants that have been identified. And then what we need to do is go back and check on the ophthalmic findings in the patient, and what we can find is that actually when you look at in detail at, for instance, the electrophysiology or other elements of the fundoscopy and so on, that actually well, yes, the patient does actually have congenital stationary night blindness. So these two things really have to marry together to make sense and make the correct diagnosis. And then you often need segregation studies and phenotypic review in family members to come to a clear-cut genetic diagnosis. And then the other thing that I want to mention is that that there are prospects for therapy now. So variant classification is very important. And I'll just give this example of this family where we found 2 variants in RPE65 and they were in trans. So meaning that one of each of the parents was a carrier for each of these variants. This is an autosomal recessive disease. And so one of them was a clear-cut pathogenic missense variant, but the other was called a variant of uncertain significance. And these sorts of variants of uncertain significance can arise in variably reported as 10 to 15% of cases when you're doing this sort of testing in eye diseases, but also many other genetic conditions. And so this puts patients in a really difficult situation, all of us really, because even though we might think they've got the clinical features of the condition, they're not eligible. They would not currently be eligible for RPE65 gene therapy because they have that variant of uncertain significance. So this was an actual case that we saw, and we noticed that this variant of uncertain significance looked to be near a splice site. So that then leads us on to other investigations that we do. This is an approach where we use stem cells for disease modelling. So this is a situation where you take blood from the patient and induce pluripotent stem cells and then differentiate them into retinal tissues and organoids. And Anai will be talking more about this later, but this is certainly something we are able to do in the lab, and Tuhar Loy in my lab has got a lot of expertise in this approach. And you can see that you can get the retinal pigment epithelium, which is the back part of the retina, and then these layers of the retina. And so we use this approach in this particular family because RPE65 is only expressed in the retinal pigment epithelium. So you can't do these sorts of studies just easily in skin from RNA from skin or blood. And so we made these RPE cells from the patients, and then we were able to see that in those cells, there was an abnormality in the splicing, which indicated a premature stop codon. So that means that that variant could be changed to a likely pathogenic variant, which is a significant factor because now the patients will be eligible for therapy. And this is work from my lab, particularly Benjamin Nash, Tuhar again, and Milan and Anai helped with making those iPS cells in the first place. And then the other strategy that we are using is using the retinal organoids for various gene replacement approaches that we wish to take. And these are also very valuable in terms of gene editing or as a model system for pharmacological approaches. So as you can see here, you get the retinal layers, and you can see in the the control situation. It's different from the disease situation, and the layers of the photoreceptors aren't there. And so then that's an excellent model system to test novel therapies. And in the lab, we have these retinal organoids in association with our mouse models to test novel therapies. So then in summary, your little overview of the where we're up to with the ocular genetic therapies in the retinal dystrophies, and all of the really critical steps in terms of the initial diagnosis, which requires the clinical phenotyping and the genomic diagnosis all working together, then with the staging of the disease to consider what's most appropriate for that particular patient. I've talked about variants of uncertain significance and how these stem cell and models can help us with those, and also how valuable they are for testing our novel therapies. And then the therapy pathways, the TGA approved one, which is also now MSAC approved for RP65 gene therapy, and the clinical trials that more and more I'm sure will be coming online, and then the novel therapies that are being developed as well. So it leaves me to thank many people in relation to this work. It's a very big teamwork that's associated with this, and including genetics, ophthalmology people, and the stem cell lab, gene therapy lab, and the funding bodies that have helped us with support this. Thanks very much. I'll now hand over to Anai, who'll talk us through the cellular therapies for retinal diseases. Thanks, Anai.
Hello, everyone. So Robyn, if you can stop sharing your screen so I can share mine. Thank you. So I'm going now to switch gears now from gene therapy to cell therapy, and just really introduce the story. A few years of research there to get to a stage where we are now very happy to move towards clinical trials using cell therapy. And when I talk about cell therapy, it's really the transplantation of photoreceptor cells in our case. So I have to start by acknowledging that this work started years ago at the Institute of Ophthalmology in London, University College London. And really, it's, as I say, it's a long story to now get to a successful outcome. And I moved to CMRI 2 years ago to establish the technology at CMRI, and also now try and develop new methodologies to set up similar schemes here. So all the work is actually based on stem cell medicine and on these beautiful retinal organoids that were introduced by Robyn just now. But they are here at the center of our program because they are models of the eye. So they're mini eyes that we generate from stem cells. And as you know, stem cells can give rise to any cell type of your body. And nowadays we have these mini niches that we can use to understand disease, develop new treatments. And in my case, we do a bunch of approach. We take a lot of different avenues to try and develop treatments. But today I'll tell you the story of cell therapy. So why are we doing that? We're doing that because the majority of the inherited retinal degenerations involve loss of photoreceptor cells. So that's why we are really interested in developing cell therapy for inherited retinal degenerations caused by loss of photoreceptor cells. And Robyn already mentioned the two main types of genetic disease in the eye: retinitis pigmentosa, causing the loss of rod cells in the periphery of your eye, and maculopathy. So these also include acquired disease such as AMD, where you're losing the cone cells at the center of your eye in that macular region that is a cone-rich area. So again, Robyn introduced the retina, but just to show you here how the healthy retina has this beautiful laminated structure composed of 6 neurons and 1 glial cell type. And again, you know, you have your light sensing cells there, the photoreceptor cells responsible for vision, the rods and the cones. And when you have a disease scenario where those cells are lost, so you pretty much lose every single photoreceptor cell in the retina, that's what we call advanced retinal degeneration. One of the only options, it's the replacement of this cell type, of course. So it's transplanting the cells back into the eye. And that's what we've been now investigating for a few years, our cell therapy, with the aim of course, rescue vision. So again, photoreceptor cell transplantation, we believe it's a very feasible treatment because the eye is an accessible structure, as Robyn explained before. It's easily accessible. It's an extension of the central nervous system that is actually outside, and you can easily inject it and reach the back of the eye where we want to transplant the cells. I say easily, but this does require very precise surgery, and we have pioneered those in the lab. So it's relatively feasible because when you lose your cells, as I'm showing here in this cartoon, and you transplant the cells back, you only need these cells, the photoreceptor cells only need to form one single synaptic connection with the next interneurons in the layer. So it's different from the brain where you transplant neurons, for example, that need to form a number of synapses and extend very long axons, for example. Here, it's a simple one cell synaptic. So this started in 2016 when with our first study where we used photoreceptor precursors that were actually isolated from mice, so very young mice, first postnatal stage. And we demonstrated that when we isolated those cells and transplanted 200,000 cells at the back of the retina, we could, these cells could survive and integrate into the retina. So that means they would connect to the endogenous retina and to a certain extent rescue pupillary responses and rescue vision. So another study 6 years later, so this was in 2012, after many years of optimizations of the whole procedure, we now transplanted a purified population of these cells. And we were able to demonstrate that indeed these cells were forming functional connections. And we also have shown that with a number of behavior tests that we could improve visual responses. So again, here we were using cells that we obtained from very young mice. That first study that I just mentioned, this Nature publication here, showed that a specific stage of development is necessary for this successful transplantation. So again, was all about understanding the development of the photoreceptor cells and isolating the specific stage for a successful outcome. This was the improvement of that technology. And what we did next, and that's where I started in the lab and led this research, was to find a renewable source of cells. So now we needed to find unlimited number of cells that we could transplant to the animals. And that's when we started our work on stem cells. This work was done with mouse embryonic stem cells. We established a protocol very similar to a pioneer study done in Japan, where they were able to form the first organoids that really mimic eye development in the dish. They would form optic cups. They would form the layers of the retina, including beautiful layers of photoreceptor cells, we adapted this protocol to generate sufficient numbers of photoreceptors in the dish for our transplantation experiments. And this is what we did then. I was very lucky that in the lab, we also have access to gene therapy. We are a very big lab in the gene therapy space in the UK and worldwide. So in this case, I use gene therapy viral vectors, AAVs, to label my cell populations in vitro prior to transplantation. So here I'm showing the organoids in the dish, and the green cells here within the organoids are rod photoreceptors that I labeled with a specific promoter, a rhodopsin promoter. So we isolate these cells using FACS and transplant these cells. Again, we transplanted 200,000 cells. I'm cutting a long story short here. We characterize these cells. We again established the specific stage of development that we needed to transplant these cells. And we're very happy to see that these cells survived in the subretinal space. In this case, we used rod transducing knockout mice. So the mice have night blindness. They don't have this rod transducing protein that is involved in the phototransduction cascade. And you can see that the green cells here, which were the transplanted cells that we generated from mouse stem cells, extended processes and express the rod transducing protein that is lacking in this animal. These cells also send processes to the other interneurons, so in this case, the bipolar cells. And when we looked very closely, they were forming synaptic buttons. So within the process, we have very clear synaptic buttons there. So we're very happy with this outcome, and it showed that we could indeed transplant cells that were generated from stem cells. Now we needed to go a step further. So we're very happy with the mouse cells, but how about human pluripotent stem cells? So always with that intention of moving towards the clinics, we wanted to now pretty much replicate the studies, but using human pluripotent stem cells. So what I did was to establish a differentiation protocol. At the time, we had a number of protocols available. You know, stem cells was, research was booming, and I needed to develop a protocol that it was relatively easy. These protocols are very laborious and demanding in terms of personnel, material, and so on. So I needed a relatively simple protocol that would generate lots and lots of retinal organoids and cells to transplant. Now, while the mouse work, the differentiation in culture took us a month to get the cells at the correct stage of development. Now in human, we're talking about weeks and months. So the first step was to really characterize the generation of these retinal organoids in vitro. And this is what I'm showing here on the left-hand side. I did a very week by week, almost week by week characterization of these retinal organoids and photoreceptor development in vitro to be able to again find that stage where the cells are prone to integrate once transplanted. So we want a photoreceptor cell that is not mature, or so they don't have outer segments, for example. Outer segments are structures that I'm showing here on the side. We can indeed generate retinal organoids with beautiful outer segments, but we don't want that stage for transplantation. We want relatively early, but we don't want very, very early where these cells are still proliferating, for example. We concluded that around 20 weeks of development, so 3.5 months in the dish, they were at the right stage for transplantation. And what we did again was to use viral vectors to label. With human cells, we actually transplanted cone photoreceptor cells because human, you know, human vision is very dependent on cone cells. So we were very interested in assessing the behavior of the transplantation of the cone cells. So we used, again, a viral vector with a promoter driving GFP only in cone cells. Very nice and strong promoter. You can see here the cells in the dish. isolated these cells and transplanted again 200,000 human cells in the subretinal space. Two different models. One model that more mimics a macula region, so it's a cone-rich model. And another model where all the photoreceptors have been lost. So it's an AAPL1 model where you pretty much lost all your layer of photoreceptor cells. So again, mimicking that advanced stage that we are likely to encounter with patients. So what we saw in this case is that, yes, the cell survived. This is 4 weeks post-transplantation in a non-immune compromised animal, okay? And we're injecting human cells. We saw that a few of these cells were able to integrate into the outer nuclear layer of that cone-rich animal that I just described, and they would indeed send some processes. I know that this is a human cell because I labeled these cells with your human nuclei. We also used antibodies specifics for human mitochondria, for example, and these cells were in very close proximity with the glial cells that actually provide sustentation to the cell mass in the subretinal space, and they also formed presynaptic, they also express presynaptic markers such as rebi shown here in this puntated staining. So of course, this again was a proof of concept study that we could transplant photoreceptors, in this case now human embryonic stem cell derived photoreceptors. But our next question was, can these cells actually connect to the interneurons and rescue vision? And that's our next work, which actually now was just published a week ago. So 2021, we started in 2016, remember? And this was the beautiful work of a very talented PhD student, Joana Ribeiro. Now she's at Stanford. And what Joana did was to first cross the mice that we had been used, so that advanced degeneration model, with an immune compromised model, because we realized that if we wanted the human cells to mature after transplantation, we needed to keep them alive in the host for more time, basically a few months, while with the mouse cells was a quick process. With the human cells, normal development just takes much longer. So she did that. We increased the number of cells. So before we were transplanting 200,000 cells, now we can transplant 500,000 cells. Remember, these mice don't have any photoreceptors left. So we can actually transplant quite a few cells. And again, now she could analyze these animals 12 weeks post-transplantation as opposed to 4 weeks. We also had a very important control that I need to introduce you. We generated an IPS line that has a CNGB3 mutation. So it's that achromatopsia mutation that Robyn mentioned before, where the cone cells are they are present, but they are non-functional. So we generated non-functional cone photoreceptor cells from embryonic stem cells, from IPS cells, sorry. So here, analysis of this study, just showing a bit of the data. We first checked for maturation of the cells 12 weeks post-transplantation. So here's just a cartoon for you to appreciate the morphology of the photoreceptor cells. And there are two features that you look when a photoreceptor cell matures. You're looking for synaptic connections and outer segment formation. So outer segment, again, is that region that will receive the light where all your opsins are located. Right? So indeed, the cell mass is there, all your transplanted cells forming a beautiful cell mass that spreads throughout the eye, and these cells could form outer segments in vivo. Remember, when we transplant these cells, they're still immature, they don't have outer segments, okay? And here we could see in both the H9, it's the cell line that we use is a wild type healthy control line usually used in these studies, and our CNGB line also formed beautiful outer segments. Next, we examined for synaptic formation, and here you see the puntated staining of typical and postsynaptic markers. Now we could see postsynaptic markers forming. Again, this animal has lost all the out of the photoreceptor cells. There's no synapses remaining there. And we also analyzed it with electron microscopy, and now we could see the typical ribbon synapses of photoreceptor cells and synaptic vesicles forming there in the eye. Again, same with the CNGB cells forming the typical synaptic connections there. Next, of course, we have to do the ultimate experiment, which is the electrophysiology and the behavior experiments. So when we did our electrophysiology experiments, we used a microelectrode array. This is basically a machine where we dissect the retina, the transplanted retina, and we put the whole retina facing down into electrodes, okay? And then we can shine lights, different intensities of lights, different wavelengths, and then assess the response to light. But the response to light, it's actually coming from retinal ganglion cells. So not from the photoreceptors themselves, but from retinal ganglion cells. So the photoreceptors have to connect with the retina so that the retinal ganglion cell responds. Okay, and here we're just showing an untreated control. The RD1 mice has no response, doesn't respond to light because it doesn't have any photoreceptors left. The wild type transplantation, so that the transplants that received wild type cones showed beautiful electrodes with high peaks in response to light. And when you have the CNGB controls, you don't see the same peaks of response. And here is just an experiment where we added drugs to really tell that this was a photoreceptor specific response. So these drugs act just on photoreceptor synapses. And again, the untreated control, it's pretty much flat. And here I just point out to some small bumps that we see on these graphs, which are due to melanopsin responsive ganglion cells. So we also have melanopsin cells in our eyes that actually have this slow response. They have opsin 4 and they have this typical slow response to light there. And while the wild type cones have this quick, actually very similar to the endogenous behavior at time 0 when light is shined. And this is abolished, completely abolished when you add the drug. And when you wash the drug out of the equipment, then you have that response back up. And again, when you transplant the non-functional cones, we don't have that quick response to light. We just see the typical melanopsin responses there. So that was very good indeed. Next, we did behavior experiments. So here we added, we used a light avoidance assay. So as you know, mice are nocturnal animals. They prefer to stay in the dark. They're more comfortable in the dark. And we have therefore built a chamber where you have a dark compartment and a light compartment. And we have an infrared camera that just records the behavior of the animal through a period of 10 to 15 minutes. And here just an image of the mice going around the arena and exploring. What we've shown in this case is that, you know, the untreated animals, so the RD1 have no preference really. So that these are the black dots here. They have no preference of what side of the arena they spend their time. The same thing with the CSGB transplanted animals. They don't have any preferences, but when we looked at the transplanted eyes, they were very similar to wild type animals that actually tend to spend 70 to 80% of their time in the dark compartment of the arena. And our transplanted cells significantly, the animals that received wild type cells significantly spend more time in the dark. And actually that's better visualized when you have this heat map here and the tracings. You can see that the transplanted H9 animals spend considerable more amount of time in the dark, similar to the wild types, and the untreated and the CNGB treated were, you know, spend same amount of time in both arenas. And also we checked for the latency, so the time that it took for them to enter the dark side, and significantly, that's also significantly different between treated and untreated. So with these experiments, we showed that we can not only rescue retinal functional, but now some basic visual functional with cell therapy using cone photoreceptor cells. So this is just a summary. And a general summary of the work is that indeed we can generate mouse and human photoreceptor cells from stem cells. We can generate retinal organoids. These retinal organoids generate sufficient number of cells for transplantation experiments. As I mentioned, we need thousands of cells. These cells can survive, they can integrate into the retina, and we can use them to treat advanced degenerations in this case. In our most recent work, we showed that we can reactivate synaptic connections with the host bipolar cells. We can restore sensitivity of the retina, and we can restore some basic visual functional, as we demonstrate with the light avoidance test. So hopefully I have convinced you that like years of research, has been more than 15 years of research now, has led us to a point that we are very happy to actually progress to clinical trials using cell therapy. And that's, as I said before, it's a major work that happened in the UK, and they are now indeed moving towards clinical trials in the near future. And we are now here at CMRI and New South Wales introducing this new technology so we can use for a number of different treatment tests of therapies. Thank you.
Thanks, Anai. Could you unshare, please?
Stop sharing.
Thank you. So look, it's my pleasure to thank both yourself and Robyn for really inspirational presentations. And we do have time for a few questions now. I do encourage anyone who hasn't fired a question in, the option is there, still there to do it. And if they can't be answered now, we can do it as a follow-up. So let me just quickly introduce our Q&A moderators, Professor Craig Munns, who's a pediatric endocrinologist and the Advanced Therapeutics lead on the Westmead campus of Sydney Children's Hospital Network, and Laura Fawcett, who's a respiratory physician and our advanced therapeutic lead on the Randwick site. So over to our moderators. Thank you.
Thank you very much, Ian. I've got the first question here from Michael Gaddis to Robyn, and it's how well do the TGA approved product or products work?
Oh, hi, Mike. Okay, so yeah, and that's a good question that that people do ask, and in fact, the TGA certainly put the up here 65 gene therapy through its paces from that perspective, and I think it's pretty clear from the clinical trial data that there is a good effect from this therapy. I can refer you to, there's a Lancet paper from 2017, and I can put this up later, from which was reporting the efficacy and safety data from the phase 3 clinical trial that happened. And certainly what patients were experiencing, and that's reported in that paper, and then subsequent clinical experience from around the world, is that they could certainly have much more mobility. So there's a luminous mobility test that happens, and you can see on those that before the therapy, the patient was really stumbling around, and then after the therapy, they're able to navigate that pathway in the dark much better. And then there's things like people can improve four lines up on the usual visual acuity measures and things like that. It can make them much more functional in terms of being able to draw and things like that, which they couldn't do. So, I mean, there's always going to be variability because these criteria need to be met. But, you know, each patient's different, of course. But I do think there's really has been very good clinical effect observed for quite a number of the patients.
Thank you, Robyn. I've got a question here for Anai. This is from Emily Ronning. So Anai, you mentioned that the whole photoreceptor layer was locked out of the animal model. Will rod cells be investigated or transplanted in the future alongside the cone cells to help rescue vision?
Yes, cones will be targeting macular degeneration. And to be honest, the majority of the work, the initial work with the mouse cells and the mouse embryonic stem cells was all performed with rod cells. It was only when we moved towards the human embryonic stem cells that we realized that it would be a good idea to start investigating the capacity of integration of the cone cells. But both cells are on our target.
Thank you. Robyn, Lisa Morgan asked if there are national databases to collect retinal gene variants.
Hi, Lisa. Yes, well, certainly we're working in that space, and we've got some funding now to work across New South Wales, and we're working on getting some federal funding as well, so that we can really be working on doing that for the patients, so that there can be accessibility for this for as many people as we can. I think that it's important, though, to be aware that it is very reliant on our usual clinical processes, because we really we can't go straight to a genetic test, a gene test with these patients. It really needs the detailed clinical ophthalmic phenotyping and analysis, and then genetic and genomic testing after that, and then often, as I said, this real interaction between the genomic side of things and then the ophthalmic side of things to work out where things are up to. So there's a lot of work that needs to happen to make this as streamlined as we can. But I think there is real impetus now with this TGA approval that we've seen come through for one of these gene therapies.
Thank you, Robyn. So Anai, this question is going to direct to you, but you might both want to jump in here. So are stem cell functional studies available clinically, and how long do they take?
Not in terms of cell transplantation, no. There are some clinical trials ongoing. Clinical trials, 6 clinical trials have actually been performed transplanting retinal pigmented epithelium, which is another cell type that also it's affected in AMD, for example, and Stargardt and other conditions. So clinical trials have been done for that cell type, and we are now moving towards, so there is a clinical trial on retinal progenitor cells ongoing in America, and but nothing yet on photoreceptor cells specifically. No, we're moving towards that.
So I think in terms of the stem cell side of things and the functional genomics models, I really think that probably does have to move to being something that's can be done in a or at least can be approved by a diagnostic setting, because I think that we're showing how valuable these model systems are in terms of making a clear-cut genetic diagnosis, and when you can see that it really makes a difference for patients, then I guess that's a real impetus to get this sort of stem cell modelling and retinal organoid modelling up and running to be helpful in the clinical space as well.
I think there's a question here that I'll paraphrase, Robyn, about, are you aware of any commercially sponsored clinical trials bringing some of these therapies then into the clinical space?
Yeah, so there are some, which are those ones that I mentioned for Stargardt and for Usher, which are actually the oral pharmacological therapies more. So, and so they're just beginning. The one for Usher that's got sites in pretty much all states of Australia. And that's an antioxidant treatment that's being investigated at the moment. And then there's another one for Stargardt, which is looking at, again, an oral therapy that is aiming to help in the ABCA4 recycling process in the retina. And that's looking at impact in teenagers. So there are those ones, and then there's certainly lots of interest in Australia now from international companies in the actual gene therapy space. So yeah, it's quite a happening space at the moment.
Thank you.
Thanks, Robyn. This question is also directed to both of you. We might get Anai to jump in first. How do you anticipate the best way to accelerate and increase the scale of the preclinical research pipeline towards the development of new therapeutics?
Yes, I guess that's what Robyn already mentioned. The use of now these human models, it's, you know, it has been a landmark path to speed up the approval. At least we hope that they are better models and they really, you know, can mimic the human condition. And I mean, as I mentioned before, and you've seen, it takes months to generate these cells, to first generate the patient cells and then grow the organoids. So we do envisage that we'll need some kind of automation in the near future, very near future, to be able to have a high throughput test of these therapies. And indeed, that's one of our main targets, working together with Robyn, Ian, everyone, to try and get better high throughput models. Robyn?
Yeah. I mean, yeah. I mean, we're constantly working on the various avenues. I think there's potential avenues through various funding mechanisms and so on that I really think that this is a space that, you know, will be very valuable for patients. And so we are focused on those sorts of areas to be able to facilitate this as much as we possibly can.
Thanks, Robyn.
I think back to you now, Ian.
Yeah, look, we've pretty much run to time. So I'd like to finish firstly by thanking our two speakers, Robyn and Anai, for what I think were fantastically interesting and inspiring presentations. I'd also like to thank all our attendees for tuning in and participating. And of course, thank you to Craig and Laura for moderating our questions and answers. So just to finish off, it's very clear, I think, to all of us that advanced therapeutics is a profoundly, rapidly evolving and exciting field. And we're going to see all sorts of innovative therapies across the whole spectrum of disease phenotypes being developed for children and adults, and many of them in the relatively near term. So please stay tuned for further exciting webinars. As I said, this is the third in an ongoing series, and we'll be notifying people of when the next one is and what the subject matter will be. So thanks again, everybody, for joining and participating, and we'll see you next time. Bye.